Today's chapter is how do organisms reproduce. In it, we will learn about DNA. First, importance of DNA copying. Second, importance of variation. Next topic is asexual reproduction. In it, we will learn about fission, fragmentation, regeneration, budding, vegetative propagation, and spore formation. Next topic is asexual reproduction. In it, we will learn about sexual reproduction in flowering plants, sexual reproduction in human beings. Next topic is puberty, male reproductive system, female reproductive system, fertilization or sexual reproduction in animal, and birth control. So guys, let's start the video.
Formation of new life from pre-existing life is called reproduction. It is essential for replacement of dead organism, continuation of life on earth. Mainly there are two types of reproduction, asexual reproduction and sexual reproduction. So first question is what is DNA? The individuals produced by reproduction are similar to each other and their parents. These similarities occurs because of DNA. So guys, what is of DNA copying? Maintenance of body design, transfer of traits, variants are introduced at the time of DNA copying. Now guys, importance of variation. Variations ensure species can survive during unfavorable conditions. For example, if there were a population of bacteria living in temperature, water, and the temperature suddenly, most of the bacteria will die. But few variants resistant to heat will survive and grow further. So guys, now starts asexual reproduction. When offspring is produced by single parents with or without the involvement of gametes formation. Modes of asexual reproduction. First is fission, second is fragmentation, third is regeneration, fourth is budding, fifth is vegetative propagation, sixth is spore formation. First, fission. A unicellular organism like bacteria splits into two or more organisms. Generally, it is of two types, binary and multiple fission. First, binary fission. In binary fission, organism splits into two new organisms under specific conditions. Example, binary fission in amoeba. At first, the nucleus divides into two. It is followed by division of cytoplasm, then finally splitting of parent cell into two daughter cells occurs. Second, multiple fission. The parent organism divides into many identical daughter organisms at the same time. Next is fragmentation, the breaking up of body of a simple multicellular organism into two or more pieces of maturing each subsequent to form a new organism is called as fragmentation. Next is regeneration. The process of getting back a full organism from its body parts is called regeneration. So guys, here is a question. What a complex multi-organism cannot give rise to a complete individual from the cutted body part. Regeneration can be used only in those organisms whose body is relatively simple when compared to organisms of its tissue or cell. But in complex multicellular organisms, cell combine to make up tissue, tissue to organ, organ to organ system, and finally organ system into organism. This is the reason that the complex multi-organism cannot form by cutted part. So guys, next topic is budding. In budding, small parts of body of parent organism grow out as bud, which detach and form a new organism. Example, Hydra yeast. So guys, next is vegetative propagation. In vegetative propagation, new plants are obtained from part of old plant without help of reproductive part. Vegetative propagation usually involves the growth and development of one bud present on the old part of plant to form a new plant. Example, bud of potato, bulb of onion. This is natural propagation. Artificial propagation of plants. A process of growing many plants from one plant by man-made method is called artificial propagation. Common method of artificial propagations are cutting. A small part of plant is removed by cutting it with a sharp knife. This is called cutting. Example, sugarcane, banana, cactus, etc. Next method is layering. A part, generally branch of plant, is bent towards ground level. Its growing tip rest is covered by soil. Example, jasmine and lemon. Next method is grafting, a method in which a stem of two plants, one with root and other without root, are joined and are allowed to grow as a single plant. Example, apple and pear, etc. Advantages of artificial propagation of plants. New plants will be exactly same as parent plants with any desired character of plants. Plants grown by the process needs less attention. Many plants can grow by same parents. Tissue culture. The production of new plants from a small piece of plant tissue or cell removed from an growing plant which is grown under susceptible medium is known as tissue culture. Advantages of tissue culture. It is a very fast technical of growing plant. Plant products are disease free. Number of plants produced in few and week of time and take less space to grow. Next topic is spore formation. In spore formation, the parent plants produce hundreds of microscopic reproductive units called spores. When the spores case of the plants burst, then the spores spread to air. When this air spores land on soil and under favorable conditions, they germinate into new plants. Example, spore formation in rhizopus.